Hey guys, Mr. P. In this video, we're going to focus on measuring the rate of photosynthesis. So we're going to look at a variety of lab techniques used to experimentally determine the rate of photosynthesis. We're also going to recall from our last videos on how the plant utilizes visible light using pigments, absorbing that light, and ultimately how that translates into increases in the rate of photosynthesis. So this particular video is going to focus on two objectives, C137 and 138. 137 talks about specific techniques used to vary concentrations of carbon dioxide, light intensity, or temperature experimentally to investigate the effects of limiting factors on the rate of photosynthesis. These are limiting factors, and they limit or govern the rate of photosynthesis, which is going to be really important if you're a plant. It is an application of skills, and so we will utilize a lot of the information presented in this video in lab, where we suggest hypothesis to investigate the effects of those limiting factors through experimentation. C138 is carbon dioxide enrichment experiments as a means of predicting future rates of photosynthesis and plant growth. We're going to specifically focus on FACE, which is free air carbon dioxide enrichment experiments, and have a discussion at the end about what those are and what the findings of those particular experiments are. So the first thing we need to look at is the photosynthetic equation. Now, in our first video in this series, we highlighted the photosynthetic equation. We related it to cell respiration, but just to recall, the photosynthetic equation reads 6 molecules of carbon dioxide plus 6 molecules of H₂O yields, in the presence of light and chlorophyll, both need to be present. Obviously, light is in the form of solar energy, and chlorophyll is the pigment that absorbs the light. Both have to be present in order for this photosynthetic reaction to take place. The two reactants turn into 1 molecule of glucose and 6 molecules of O2. Now, the two reactants are going to be used during photosynthesis, and the two products are going to be produced. And so if we think about that, and we think about how we can try to measure something to determine the rate of photosynthesis, we can focus on the usage or the consumption of carbon dioxide. And so carbon dioxide, being a reactant, will be used during photosynthesis, used being the key word. CO2 concentration will decrease as the photosynthetic process progresses. O2, over here being a reactant, will actually be produced during photosynthesis. And so as photosynthesis progresses, the O2 concentration will increase. If we increase the rate of photosynthesis, the rate of O2 production will increase, and the rate of CO2 usage will increase as well, which means the concentration of carbon dioxide will actually fall because more and more of the CO2 is being used. If the rate of photosynthesis overall decreases, you would expect to see a decrease in the usage of carbon dioxide and also a decrease in the production of O2. So these two, being the products, are going to be produced during the reaction, and these two, being the reactants, are actually going to be used during the process as well. We can focus on looking at the consumption of the reactants, or we can look at the production of the products. Photosynthesis and cell respiration are both players in a plant's cellular energetic pathway. Photosynthesis is obviously the production of glucose, and cell respiration would be the usage of glucose to turn glucose into ATP. So photosynthesis essentially takes CO2 and water, turns it into glucose as well as oxygen, and then cell respiration takes that glucose and turns it directly into ATP, which is adenosine triphosphate, and that is the usable form of energy for a plant and or other heterotroph. So there is definitely a relationship between photosynthesis and cell respiration. And so if we look at this particular graph, you'll notice that we have a baseline. That baseline indicates neutrality, or is a neutral point, meaning the amount of O2 produced equals the amount of O2 taken in. We know that photosynthesis is an O2 producer, and we know that cell respiration is an O2 user. And so if during the day, when light is present, you would expect that O2 is being given off in a positive manner, meaning more oxygen is being given off than is being used. Because throughout the day, a plant is going through this cell respiration pathway as well. It's just using photosynthesis, or you could say that photosynthesis is outproducing the O2 that cell respiration is using during the day. So O2 is given off, O2 is given off, O2 is given off. The plant is using photosynthesis. And when the light is turned off, meaning it's nighttime, the plant switches from photosynthesis into a cell respiration pathway, meaning O2 is now being taken in, or O2 is being used, not produced. And so you see that we actually kind of go up into a positive O2 production, and then we go negative into an O2 usage. And a plant will continue to cycle as long as light is available during the day and not available during the night. So photosynthesis rate peaks during the day when temperatures and light intensity are high. And that's an important distinction. Not only is light intensity the highest during the day, typically temperatures are the highest during the day as well. Light intensity and temperature are both limiting factors for a particular plant. As light intensity increases, the rate of photosynthesis increases as well. As temperature increases, the enzymes that are used or employed during photosynthesis and cell respiration actually will work more efficiently to progress the plant through photosynthesis. Photosynthesis rate drops during the night when temperatures and light intensity are low. Again, lowered temperatures and lower light intensity are going to decrease the rate of photosynthesis, limiting factors. So what are some experimental methods that we can use in a lab setting to measure the rate of photosynthesis? The first is a photosynthometer. A photosynthometer is a type of apparatus, or is a lab setup, that can be used to measure the rate of photosynthesis with various independent variables. We can obviously increase the temperature of our water bath. We can decrease the temperature of our water bath. We can measure the temperature of the water bath with a thermometer to ensure that we're holding it consistent, because it is important for us to control independent variables and to control other variables as well. We can vary the light intensity. We can take a light source and we can move the light source closer or farther away. We could put a mesh screen on it. We could take light totally away. We could change the color of light. We could shine red light exclusively. We could shine blue light exclusively. We could shine green light exclusively. And so not only does temperature get manipulated, light intensity, light color can be manipulated as well. Carbon dioxide concentration can be manipulated too, because when we use these aquatic plants, we typically put them into a solution containing sodium hydrogen carbonate solution, which is a carbon source. And so if we increase the carbon concentration of that solution, that would be indicative of a higher carbon dioxide concentration, and conversely, we could decrease the carbon source of that particular solution. So we can manipulate carbon dioxide concentration. We can manipulate all of these variables, but ultimately what we are measuring is the oxygen gas that is being collected as a byproduct of photosynthesis, because as this plant photosynthesizes, it is producing oxygen gas, which can actually be contained within this tubing and can be collected with this syringe, and these syringes will have markings or gradations, and we can actually measure the amount of oxygen gas given off. We can determine the rate of photosynthesis based on the amount of oxygen collected. If we increase light intensity, we would expect to see a higher rate of photosynthesis because of a higher output of oxygen gas. If we lower the temperature, we would expect to see a decrease in photosynthetic rate and a decrease in the quantity of oxygen being produced. If we decreased our carbon dioxide concentration, we would expect to see a decrease in the oxygen gas being collected. All of these are limiting factors, and this is a really good way of manipulating those variables and then measuring the oxygen gas being produced and relating that to the rate of photosynthesis. Another experimental method for measuring the rate of photosynthesis would be using an aquatic plant and not collecting the oxygen gas, but just simply counting the bubbles. So when an aquatic plant is put into an aqueous solution within this beaker and shined a light on, it will go through photosynthesis, and so we can simply count the oxygen bubbles that are given off. As we, for instance, increase the distance of this light source from our plant, we would expect to see the bubble production rate to drop, meaning the plant will produce fewer bubbles. This might look like 10 bubbles per minute, it might look like 20 bubbles per minute, it might look like 5 bubbles per second, it might look like 1 bubble per second. You will count the number of bubbles, that will be the quantity, and you will put it over the amount of time you're collecting data for, and that determines a rate. If we decrease the distance of this light source, we would obviously expect the light intensity to go up, and therefore the bubbles being produced go up as well. Light intensity can be manipulated using a ruler. That would be our independent variable in this case. We could also change the light intensity by keeping it at a constant distance, but just manipulating the amount of light reaching by putting a screen, or you could manipulate the color of the light in this kind of experimental setup as well. Bubble number per unit of time can be recorded, and the volume of oxygen produced can be recorded as well if you were actually collecting the gas that is given off. Another experimental method would be to use indicators. So these are hydrogen carbonate indicators. You will see that there is an indicator added to our solution, and it will change color depending on the pH. This scale runs from about a pH of 7.6 to a pH of 9.2. Obviously, this would be the more basic end of that pH range, and this would be the more acidic end of that pH range. And the reason we use these indicators is because CO2, when in solution, is going to change into carbonic acid. And so carbon dioxide in water produces carbonic acid, which reduces the solution pH. As CO2 concentration increases, it's going to cause the color to shift more to this yellow range. And if CO2 concentration decreases, it's going to cause the shift to the purple range. And so when we shine a light onto our samples, these would be algae beads, which would be beads that are locked in sodium alginate. We put them into a solution containing these indicators, and as we shine light on it, whether it's colored light, or we change light intensity, or we change temperature, or we change the carbon dioxide concentration inside, you will actually start to see, based on the rate of photosynthesis, a color change. And so if you change the light intensity and put our algae beads into these vials that contain this indicator, and photosynthesis is working really well, CO2 would be decreasing and therefore would be turning the vial more purple. If photosynthesis isn't working very well, or if cell respiration is outpacing photosynthesis, you would expect CO2 to be produced, and that would cause the indicator to shift more to this higher CO2 indicator range, which would be yellow. As carbon dioxide is used during photosynthesis, the solution pH will rise and the resulting color change can be recorded. Again, higher levels or higher rates of photosynthesis would be indicated by a darker purple color, because it would be using the CO2, which is pulling it out of our solution, which is decreasing the CO2 in our vial. And the last and probably easiest method for measuring the rate of photosynthesis would be simply using digital probes. These digital probes are sensors that can be used to measure the amount of carbon dioxide and or oxygen molecules in carefully controlled atmospheric settings. So you might have leaves that you are shining a light on, you might be manipulating the light intensity, you might be manipulating temperature, you might be manipulating the color of light. But you often will have a CO2 sensor and an O2 sensor, and that will basically tell us how much CO2 is being used by the plant with a dropping CO2 concentration and how much O2 is being given off. If CO2 is dropping more quickly, you would expect the rate of photosynthesis to be increasing, and if O2 is being produced more quickly, you would expect, again, the rate of photosynthesis to be occurring really quickly. So these are all different experimental methods used to measure the rate of photosynthesis. We will do versions of these in class. To kind of pull all of this together, there are a variety of limiting factors that have been talked about through this video, and so to summarize all of them, light intensity is obviously a really big limiting factor. Light intensity talks about how much light is actually being given to the plant. We also have amount of water, which we haven't really talked about in this video, but the amount of water available to a plant is definitely a limiting factor, because if you remember, CO₂ plus H₂O yields, in the presence of light and chlorophyll, C6H12O6 plus 6O2. Water would be a limiting factor because it is a reactant in this pathway. CO₂ concentration would be a reactant in this pathway as well. Temperature is a big one. If temperature goes up, you would expect the rate of photosynthesis to go up, and if the temperature goes down, you would expect the rate of photosynthesis to decrease. That has everything to do to the enzymes and the proteins that are in the cells of these plant tissues. As temperature goes up, the enzymes work better because of those molecular collisions. If temperature goes down, the enzymes don't work as efficiently, they just slow down, molecular collisions decrease, and so temperature is definitely tied as a limiting factor to the rate of photosynthesis. Like I said, carbon dioxide is a reactant, and so carbon dioxide will definitely alter the rate of photosynthesis. Typically, higher carbon dioxide concentrations will result in an increase in photosynthetic rate. Chloroplasts and chlorophyll. Like I said, chlorophyll is one of those things that must be present. The chlorophyll is the pigment, and the pigment is contained within the organelle, which is the chloroplast. And so both of those, the concentrations of chlorophyll and the concentrations of chloroplasts within a plant, are definitely a limiting factor. If a plant has a lot of chloroplasts and therefore a lot of chlorophyll, they're going to be more efficient at actually increasing their rate of photosynthesis, and conversely, if they don't have a lot of chlorophyll or a lot of chloroplasts, they will not photosynthesize very well, even if all of these other limiting factors are in really good concentrations. So, looking at three specific limiting factors, light intensity, carbon dioxide concentration, and temperature, let's look at how each of these three big limiting factors affects the rate of photosynthesis. When we look at light intensity, you will see that as light intensity increases, which is on our X, the rate of photosynthesis increases quickly because higher light intensity increases the rate of photosynthesis, but you'll see that it quickly peaks, and the plateau means that the enzymes that are responsible for navigating photosynthesis are already working at their maximum rate. So once they reach their maximum efficiency, increasing the light intensity won't make them work harder or better or more efficiently. Enzymes have a maximum work rate, and once you reach that maximum work rate, it will plateau. Even despite increasing the light intensity, you can't just continuously increase light intensity on a plant and expect the rate of photosynthesis to continue to increase. There is a maximum rate. When we look at carbon dioxide concentration, again, that graph looks very similar to the light intensity graph because as we increase carbon dioxide concentration, which again is on our X, the enzymes that utilize carbon dioxide, like Rubisco, are again going to be working at a maximum efficiency. And so we plateau. You cannot continue to increase the carbon dioxide concentration and expect the plant to continue to increase the photosynthetic rate. If you look at the temperature graph, this is the graph that looks different than the other two. Because temperature is tied directly to protein structure and enzymes are proteins, there is an optimum temperature that all enzymes work at. And so the optimum temperature will be the temperature that the rate of photosynthesis peaks at. As we increase the temp to optimum, the photosynthetic rate will increase to a maximum rate. And then if we keep increasing past our optimum, you will actually see a pretty quick decrease in the rate of photosynthesis. And that's because our proteins become denatured, which means the three-dimensional conformation of our protein starts breaking down. Those hydrogen bonds and disulfide bridges break, and our protein gets pulled apart and therefore can't work correctly. And if our enzymes aren't working correctly, they're not going to be able to utilize the carbon dioxide or the light at its optimum efficiency, and so it will drop to virtually zero, which means our proteins are completely destroyed. And so what does that look like In a real-world experimental setting, we have what we call FACE, free air carbon dioxide experiments. And these experiments allow us to examine the effects of increased carbon dioxide levels on plants and in natural and agricultural ecosystems. By artificially increasing the carbon dioxide levels in these natural systems, we gain a more reliable picture of what may happen in the real-world future of steadily increasing carbon dioxide levels. So the reality of the situation is our carbon dioxide concentrations are increasing and have been increasing for a long time. FACE experiments, or these free air CO₂ enrichment experiments, are large-scale ecological studies used to understand how increased levels of carbon dioxide, CO₂, affect plant growth, physiology, and ecosystems. These experiments are designed to simulate future atmospheric CO₂ conditions in natural or agricultural environments. And so how these experiments work is that there are typically large circular plots that are typically between 10 and 30 meters in diameter. These are established in fields or forests. Vertical pipes or towers are going to be put around that plot to surround the plot, and they constantly release CO₂ gas to increase the local concentration of CO₂ within that circle. Sensors continuously monitor wind speed and direction to adjust the CO₂, release more or uniform CO₂ concentrations to ensure that there is a uniform distribution of CO₂ to the plants within that perimeter. And they compare those plots to similar plots around that same site without the CO₂ enrichment, which serve as controls to compare the effects of the plants in and outside of those plots. And these experiments have led to a lot of key findings, and some of those findings are increased photosynthesis, meaning higher CO₂ levels enhance carbon fixation in C3 plants. These plants are like wheat, rice, soybeans, which would be a benefit to agricultural settings because it does show that higher CO₂ levels have increased the carbon fixation in those plants. Some plants grow faster while others show limited response due to nutrient or water limitations. Again, CO₂ concentration is not the only limiting variable or the limiting factor within plants, and these experiments are showing that. Just because we increase one limiting factor doesn't mean we're going to increase the rate of photosynthesis, because there are other limiting factors as well. Some crops have reduced protein and mineral content under elevated CO₂, and altered interactions between plants and soil microbes and herbivores just because of the increase or decrease in the nutrient availability and nutrient dilution of those particular plants. These are really important experiments that are going on right now, and they're helping to predict future crop yields and food scarcity. They allow us to assess how forests and ecosystems will store carbon long term, and they really are informing climate change models and agricultural practices, because we need to kind of stay at the forefront of what the planet is doing, and part of what the planet is doing is the CO₂ concentrations are increasing. So that's it for this video. If you learned something, give it a thumbs up, leave any question you have in the comments, subscribe to the channel. We'll see ya.